In 1928, Alexander Fleming discovered penicillin. In 1955, the polio vaccine was invented. Yet, it wasn't until 1990 that humans truly began to discover who they were. The entirety of our genetic makeup had barely been touched. The journey of studying human genetics began with the discovery of the double helical structure of DNA by Watson and Crick in 1953. With continued research and discoveries, scientific leaders began to realize the significance of mapping the complete set of genes, otherwise known as the sequencing of the genome.
People in the past have always considered the human genome as the book of life, or a set of instructions that provides guidance or information to the cell of, how to function or how to develop early in life.
In 1990, the Human Genome Project, funded by the Department of Energy and the National Institutes of Health, officially launched under the director James Watson and later Francis Collins in 1993. Its goal? To identify all of the genes that make up the human genome.
The Human Genome Project has led to significant improvements in DNA sequencing technologies, advances in the diagnosis and treatment of diseases, and emphasized ethics in biomedical research. Thus, the Human Genome Project has pushed the frontiers in medicine and science by allowing new opportunities in the biological world. The Human Genome Project was often looked down upon in its earlier endeavors. The project's projected budget of $3 billion and its unusually large collaborative efforts raised several concerns in the scientific community.
The Human Genome Project was overwhelmingly unique, first of its kind in biology, to not only get lots of people together in one country, but to do it across the whole world. I mean, there was like 6 or 7 or 8 countries involved in the Human Genome Project. That was unbelievably unprecedented and unbelievably complicated.
There was also the issue of ethical barriers.
Genetic information, especially when it has clinical relevance, it should be yours and not anybody else's. And any technology can be used to do something wrong. And I think the biggest wrong here is not having genetic privacy.
It was recognized that genes are an incredibly integral part of someone's identity and that
there probably is nothing more intimate about each of us than our blueprint. It's really, there are other things that are very personal, but you can't get more personal than your unique blueprint.
the Human Genome Project was constantly adapting to modern confidentiality standards and altering their project to fit within the ranges of consent. Many of these contentions only magnified with changing privacy norms. Despite these technical frontiers, it was an ambitious project that incited competition. A new private company began pursuing similar objectives, captivating the whole world in their race to sequence the human genome. Celera Genomics, founded by former HGP member Craig Venter, emerged as a rival to accelerate genome sequencing. As this race was covered by the press, the public began to see significant differences between the two teams, especially regarding their access to research data. The Human Genome Project worked by the philosophy of the Bermuda Principles, which allowed for public access of DNA sequencing data, while Celera only gave out this information for a fee.
Most scientists don't generally share their information straight away. They kind of like to discover something and then write a manuscript about it, and then they will share it afterwards. So we kind of broke that model in the Genome Project, and we all got together in a place called Bermuda, and we discussed the merits of sharing data straight away, and we decided that this data should be shared very freely.
Despite, or possibly due to the rivalry, both teams promptly finished their first working draft of the genome in 2000.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. But today's historic achievement is only a starting point. There is much hard work yet to be done.
Shortly after, Celera began to decline. Its subscription-based model was not gaining enough revenue, and critics began to see inaccuracies within their sequence. Thus, in the race to complete the best working draft, the Human Genome Project emerged as the ultimate victor when they produced a more precise draft of the genome in 2003. This race led to larger questions regarding public and private sector genomics. The Human Genome Project, in the public sector with other government-funded organizations, focused on a meticulous process that yielded good results. Oppositely, Celera Genomics was more profit-oriented, as most private sector businesses are. In the end, a general consensus was that the Human Genome Project won the race, as they were willing to share this fundamental research globally.
With the end of the race and the finishing of the genome sequence, the world was finally able to receive the blueprint of a human being. The resolution of the Human Genome Project was just the beginning of a new era in the world of medical science. Notable frontiers were advanced, especially in cancer diagnosis and treatment. With the creation of the Cancer Genome Atlas, a database of cancer-based genetic data, many scientists have been able to develop personalized medicine and cancer therapies. The treatments generated from this genetic information have the potential to diagnose a patient in seconds, as well as suggest targeted treatments based on the patient's DNA. The Human Genome Project has also allowed for patients to be diagnosed with possible diseases before physical symptoms arise. Medical professionals can then prescribe medications based on the patient's genomic information to optimize medical treatments and avoid adverse reactions.
Massive, growth since the Human Genome Project has really been this expanse of trying to identify differences, or what we call in the field variants, that can be associated with human disease and try to target new therapies and new therapeutic products.
Along with these significant advances in diagnosis and treatment of diseases, the project's impact on DNA analysis technology cannot go unrecognized. When the Human Genome Project began, DNA sequencing machines were rare and most sequencing had to be done by hand. This method proved impossible in sequencing the 3.2 billion letters of the human genome. Thus, a large-scale method of DNA sequencing was required. This posed a frontier in the sequencing process, as the project was forced to decrease the size but maximize capabilities of their machines. With the progression of the project, automation became more cost-efficient while also minimizing coding errors, proving the major benefits in the cost and accuracy of DNA sequencing.
When the Genome Project began, it was way too expensive to sequence DNA to actually sequence the human genome. So we had to figure out how to really decrease the cost, even to sequence one human genome, which is what the Human Genome Project did. The first sequence of a human genome cost about $1 billion, roughly. That was what the Genome Project did, $1 billion. Now we can sequence any one of your genomes for less than $1,000. So in the last 20 years, since the end of the Human Genome Project, we've done incredible things technologically that now allow us to routinely sequence the human genome very inexpensively.
As genomics becomes more integrated into our everyday lives, ethical, legal, and social implications will continue to arise. Issues in genetic testing have only revealed the importance of ensuring responsible use of genetic information. From medical treatments to DNA sequencing, advances in medicine and science still advance frontiers and will continuously do so, transforming the healthcare industry and changing the lives of countless individuals for the better.